Okay, good morning, everyone. So today we'll be talking about rates in biology. And just to point out, we're reaching the end of the course, surprisingly. This is the next to last formal lecture. The next lecture will be about information and errors in biology. And just to set us all straight in terms of the dates, so the last exercise will be posted just when I get back to the office. So that will be the last exercise in terms of things you have to solve. You'll have there an interesting question, I think, about ribosome polymerization and what it suggests about the growth rate of cells. And then you'll be given the draft of Chapter 4 about rates and durations in biology, and you should choose a vignette of your interest and write half a page about it, just like you did before. That should be handed next week, and next week you also, by then, you need to send me the subjects of your final assignment that you've chosen. You can think outside of the box. I'm open to any suggestion. If it wouldn't be proper, I will let you know, but I'm guessing it will be, everything should work out. And we'll start the student presentations in 2 weeks, okay? And I imagine it will take us 1 or 2 sessions. So that would be like today is lecture 5, next week is the last lecture, lecture 6, and then lecture 7, maybe lecture 8 will be actually you speaking in front of the class. Oh, well, any questions on that? Right. Yes. So thanks for that. So the due date for the assignment would be when you're giving the, or actually it might be, let's do it, let's say 2 days later. So if you have interesting feedback from the class, you can still incorporate it into the vignette. Okay. So I'll send also information exactly on that. Thank you. Okay. So to kick us off, we'll be talking about rates. So let's start with one of the most fundamental processes and ask ourselves, what is faster? Let's say in bacteria, transcription or translation. So this is like the famous derby of the central dogma of biology, fighting off who's better or faster, transcription or translation. And the answers would be, well, transcription, B would be translation, and C would be roughly the same. For those of you who remember that one pager on key numbers in biology, that probably could help in trying to figure things out about this. So who's voting for A, transcription? About half the class. Translation, almost the other half. Roughly the same. We're getting 1 or 2. Okay, excellent. So let's see how are we handling it. So what do we have involved? So let's start with the DNA. I'm not very good at depicting this. Let's just do it this way. This will be the DNA. On top of the DNA, I'll have my RNA polymerase. And this would be performing transcription at some velocity that I'll be interested in. Out of that would come out my transcript. So if this is going this way, it would come out, let's say, like this. And on top of that, I'll be having the ribosome. And the ribosome will have another velocity. Let's call it, this would be the velocity of the ribosome. And this would be the velocity of the polymerase. So what do we, yeah? Okay, so the question is about the initiation. So I'm focusing here only on the elongation steps, on the transcription and translation in that respect, and we can then go back and also discuss initiation. Good. Okay, so what?
So obviously you're talking about bacteria.
I'm talking about bacteria in the way I plotted it, because we're thinking about concurrent transcription and translation, and we'll talk about what happens in eukaryotes afterwards. Okay, so these two values are quite critical, and that's again where, having some number, I'll be, if you never thought about these numbers, it's hard to deduce them from thin air, but these are some of the really very key numbers in molecular biology. If you know these numbers, you can calculate lots of other things like you already experienced, and therefore it's worth trying to remember them. So what do we know about the rate of polymerization by the RNA polymerase? Anyone has a number off the top of their head? So it's several tens of nucleotides per second. So this is roughly, let's say, 30. Excuse me. Let's say, I think we wrote it in the key numbers is 40 to 80 nucleotides per second. Okay, NTs for nucleotides. And this would be the characteristic numbers I'm giving here. So in biology, everything can change quite roughly by more than a factor of 2. This is what has been measured for being like a maximal rate of transcription in eukaryotic cells, say in E. coli, more specifically. Okay, the actual values could be somewhat slower. We could talk about what happens in eukaryotes. Let's compare it in terms of what happens under the most favorable conditions. Okay, no special hindering or Scheindel-Garno stopping you in all sorts of different ways. Okay, so this is about the rate of the polymerase. Then talking about the rate of the ribosomes. Rate of the ribosome in E. coli is roughly 10 to 20 amino acids per second. So now in order to put them on the same footing, we just have to consider what happens in terms of amino acids per nucleotides. So for every, I have to multiply by 3. For every amino acid, effectively 3 nucleotides. So we're talking about here something like 30 to 60 nucleotides per second. What? Right, so we'll be talking about that in a minute as well. So just to answer, so going back to this question, I'll say they're roughly the same, okay? I'm not looking at small differences here because there's quite a lot of variability, but they're on the same order of magnitude in terms of rates in bacteria. Let's think what could it have been different? So what possibilities do we have? It could be either that transcription was faster or translation was faster, right? So what would happen if translation was faster? There's actually a problem, right? Because if we're talking about bacteria in which translation starts just as while transcription is still being done, if translation was faster, the ribosome would bump into the RNA polymerase. So we're limited there in any case. Actually, there are now all sorts of studies, really interesting studies where people are able to measure more and more, and they're starting to see that actually often the ribosome might even be sitting just after and actually helping to push things forward to some extent. So there's interesting things coming out about that. So we see there's an issue about the rate of translation being faster. What about transcription?
About the last point.
Yeah.
But a single RNA strand can work in a few, it's not a one-time thing. It works for a few times of translation.
Right. So it wouldn't be one ribosome. You'll have several ribosomes sitting here. This is what's known as a polysome. And even after the rate of transcription has ended, you'll still be having the transcript. And then you're saying you could have had the ribosomes moving even faster.
Yeah.
Yes, I agree. After the initial rate of transcription, you could have things potentially working faster than that. And then if transcription was faster than translation, well, that could happen. Then, for example, you could have, for example, issues of degradation, because now you have the nascent transcript. It's open for all sorts of RNases that could start degrading it. And maybe there are mechanisms to handle that, maybe not. I would say that for me, it's an interesting question of not only what is faster, but also what governs that characteristic rate. Why isn't it, are there any limitations on having things working, say, 10-fold faster, both transcription and translation? If anyone has a good answer, I'll be delighted to hear it. It's a good subject for a final assignment or maybe for your PhD thesis. I think it's a pretty deep question. As you'll see from the exercise that you'll be doing about the ribosome polymerization rate, how it affects the growth rate and the proteome in what you'll be doing as an assignment for next week, you'll see that if we were able to do things much faster, that could have quite significant implications. So it's, for me, an interesting puzzle on what governs that rate. Okay, if we're talking about, so we were talking up till now about bacteria. So if we're talking about eukaryotes, so in eukaryotes you have a separation between the two processes. The transcription will happen in the nucleus, and then you'll have an export of the mRNA outside, and then you'll have translation only happening there. So if one looks at the characteristic rates for eukaryotes, they wouldn't be so far from the numbers that we see here. They'll be, say, the rate of the ribosome could be several amino acids per second. There's been recent measurements by the group of Jonathan Weizmann, where I think about 8 amino acids per second in mammalian cell lines. The rate of the polymerase could be a few times, say, about 10 nucleotides per second, but we're talking in the same ballpark, just somewhat slower. And also still in the same order of magnitude between the two processes, even though now they're separated in time. But there is actually one significant difference if we're thinking about the rates where the overall transcription and translation would take place between eukaryotes and prokaryotes. Anyone sees the major difference? What?
The export of the RNA.
So there is the export process, that's true.
But it's private.
Right, so if we're thinking about what happens in eukaryote in terms of transcription, you'll also be having the introns. So the length of the introns could be, on the average, is about 30 times longer, say in human cells, the average size of intron than the average size of exons. So even though the rate per nucleotide would be roughly the same, the overall time would be quite different. And we were talking here only about polymerization. As was mentioned here before, there's also the initiation process that we're not taking into account here. And sometimes the initiation could take as long as the polymerization itself. And we'll be mentioning that later on as well. So starting from that, let's move to a follow-up question that I think is quite related. And that is, how long would it take to make a functional protein, let's say starting from having the gene on the DNA? So we have the relevant numbers at hand already here. So what is the length of a characteristic gene? Right, so the length of a gene would be on the order of 1 kilobase per, say for bacteria, talking about the expressed part. And therefore, if we think about the time, so, and this is roughly 300 amino acids. So if I'm thinking about the time that it would take to transcribe, or let's talk about translating it, it would be about 300 amino acids divided by the rate of the ribosome, so let's say about 10 amino acids per second. So it's about half a minute. So when thinking, so you could have the transcription and translation could be coupled. So basically, if you need an order, like a rule of thumb about possible rates of production of a gene, it would take on the order of a minute. And as we said, if we're thinking about transcription in eukaryotes, it could be significantly longer because of the introns, because the length now would not be 1 kilobase pair, but could be 30 kilobase pairs, even if you have, in the end, just 300 amino acids that would need to be translated. So this tells you something about, say, if you have a cell and you put your plasmid or some gene of interest, and now you're inducing it, let's say, with IPTG, and you were able to look under the microscope and to see in single molecule resolution, how long would it take until you see the first sign of something being expressed, right? There are all sorts of caveats involved in that. One is, for example, if you're looking, say you're expressing GFP or some fluorescent protein, you now have a delay coming from what? Maturation process. So fluorescent proteins have a maturation process that relates to all sorts of oxidation, all sorts of cyclization of the molecule, and that could be, depending on the molecule, could take several good minutes. Okay, so that would be one thing to take into account. But also, we're talking here about what it would take in terms of timescale to give you, say, the first functional protein. Yeah, a minute or just a few minutes. This does not say when will you be getting, say, half of your steady state amount, or how long will it take until you reach the steady state amount. So let's talk about that distinction for a minute, okay? Let's try and make that a bit clearer. So we have a cell, we're starting to, say, induce something. If we think about the dynamics of the level of the, say, I have a cell, at some point I'm inducing it, and I'm interested to know what's the concentration as a function of time of, say, my protein of interest. Okay, or you can think about it also as the mRNA level. I'm talking about quite generic equations here. So one could write a differential equation in which the concentration changes with time, and that's what we are interested in. It would have some synthesis rate that we'll be denoting by beta, which is the rate at which things would tend to accumulate just because we started to produce something. So you could think about it. I chose C so as to be somewhat agnostic to that. You can think about it. I think it would be easiest maybe to think about it as mRNA, just for simplicity, because it's not two stages. If there are two stages, you need to think about separation of time scale. Forget it. Let's just think about it as mRNA. And what do you have except for the fact that you're producing? You also have degradation. And there's actually, so let's write it. I'll write this alpha times the concentration. Why? Because the more I have, the more I'll be losing to degradation, right? And there's a fine point here that I'll try to explain, but not in too much detail. In thinking about alpha, one issue that relates to that is, if I'm thinking about what is alpha, it's composed of the rate of degradation. But there's another component here, because I will think about concentration. I'm not talking about the absolute number. I'm talking about the concentration. The concentration of the molecule could change by two things, or could be lost if you want, or decrease by two things. One is, as was pointed out, by active degradation of the molecule. What else could decrease the amount of protein that I'll have, or mRNA that I have inside the cell, the concentration of growth? That is known as dilution. Okay, so I had here degradation. I also have another effect, alpha dil, that relates to dilution. From the fact that if everything else, if the cell grows and I'm not producing anything, it's effectively like degrading, or I'm having the concentration going slower and slower.
Also can be activated like glucose once entering the cell to reduce the amount of glucose. It will be phosphorylated. So immediately moving from one food to another.
Right, so you could also have the concentration changing by the fact that it's not degrading, it's not diluting. It's actually you have some process in some assembly line or something and it's being turned into something else. That's also true. Yeah. Okay, so we have here a combination of degradation and dilution that governs the rate in this equation. Okay, where should we go? Okay, so now let's try and plot how we expect the temporal evolution of the concentration to look like. So let's say that I'm starting at time 0, and that's where I've added my IPTG, or whatever inducer. And I'm further assuming that at time 0, I had, a negligible amount, although you could have some basal amount. So as we were discussing initially, we won't have any response because we have some delay, if you wish, that comes from this order of like a minute. Maybe it could be somewhat longer because we were taking maximal rates, so it could be very few minutes. Then we'll start to have accumulation, starting first with a slope that depends on beta. And then this would saturate into some steady state level. What would be the steady state level? So that's not so difficult to calculate if I want to know the level that I'm going to get at steady state, because at steady state, this would be equal to 0, right? Because steady state means that the concentration does not change with time. Now I'm interested in the value of C at steady state. So you could just change sides and you'll get this value being beta over alpha. But what I'm most interested in is about the dynamics involved. And therefore, I'm interested in this time scale. The delay in the response. Now, if I really want to get to the value of beta over alpha, that would actually take a very long time. That would be, the final bit of getting there would take quite a while. So usually it's good to think about, the functional level could be some arbitrary factor on this. Let's say the time until it reaches, say, half of the maximal level. So say beta over 2 alpha. And that's an operational way to define the time scale I'm interested in of the delay. So this value for the delay, it's not so complicated to solve it, but I'm sure you might be doing it in other courses. I won't be doing it exactly here. Let's just go directly to the answer. So the time of delay goes as ln 2 over alpha. And anyone who wants to see exactly how this is being done and explain much better than I can do should go and check out the book Introduction to Systems Biology, which has lots of good, fun and interesting stuff, and also very good explanations by Uri Alon, our very own Uri Alon from the Weizmann Institute. So that's highly recommended. You can see all sorts of interesting explanations and twists also on this dynamics. We're talking here only on what happens when you don't have very special regulation, but there you can also discuss what happens if you have positive regulation, negative order regulation, all sorts of more advanced processes.
Delay is from the beginning of the time or the beginning of the rise?
I beg your pardon?
The delay is from the beginning of the time, the tau at the beginning
Yes, I was, on purpose, I was a bit unclear here. So you first have some sort of a delay inherent that we were talking about a minute, but then you have this delay. So and this would be on top of the 1 minute or whatever response time. But let's talk about what would govern. So, alpha does not help. You're interested in probably, in the spirit of this course, we're interested in specific concrete numbers. So let's try and discuss what would govern this alpha. So this would be, to a good approximation, depending on 2 issues. in a way, you could say this would be like the minimum of the time of degradation and the time of the cell cycle. Where did these two things come from? It came from the fact that the value of this alpha depends on two processes, degradation and dilution. From each of these processes, I could get a time scale. From the active degradation, I'm getting some characteristic time of degradation. We'll be talking about the numbers in just a second. On the other hand, from dilution, you'll also be getting a time scale. What is the time scale related to dilution? That's exactly the one related to growth. That's the cell cycle. And therefore, the response time here will depend on, you'll either be, the time that it will take you to respond either would depend on the degradation or on the cell cycle. Let's try to make it a bit more concrete. Let's think about what happens, let's say, in bacteria, in yeast. Let's say I'm talking about budding yeast, and in a mammalian cell line. Let's say HeLa. And I'll be thinking about mRNA and protein. So I'll be taking a risk here, if you allow me. I'm trying to make, broad generalizations on all these, different model organisms, very different characteristic biological processes. But I think it's worthwhile to do that. And obviously, there could be some differences on the specific case. So bacteria mRNA. So I'm asking about what would govern the response time, the delay, in thinking about mRNA accumulation in bacteria after I use some inducer or repressor or whatever. So anyone knows something about, so what would be faster? What would be the shorter time scale? We're interested in the shorter time scale. Would it be degradation or would it be the cell cycle? For bacteria. So what's the cell cycle for bacteria? A few, yeah, so the fastest would be, say, 20 minutes. But actually, if you look at the mRNA degradation in bacteria, characteristic number, the average number or the median value for E. coli would be roughly 3 minutes. So mRNA for bacteria would be dependent on degradation of the mRNA. And actually, this could be generalized. In most cases, mRNA would be dominated. The shortest time scale would be degradation. And the average values or characteristic values would be 3 minutes for yeast, 3 minutes for bacteria, 30 minutes for yeast, and say about 300 minutes or a few hours for HeLa cells. Just a second. On the other hand, if I'm looking at proteins degradation would usually be, and we were calculating some estimates on that, if you remember in the previous lectures on energies, would usually be slower than the cell cycle. So here, the governing time scale would tend to be the cell cycle. So if we want to make things nice and round, you could say, let's say this is 30 minutes, this is 300 minutes, a few hours, and this would be, this could be 3 hours, and this could be 30 hours. Okay, characteristic mammalian cell line is on the order of a day.
You're talking about steady state. I mean, like.
So I'm talking about exponential, things in exponential. Yeah, so I'm talking about here, so mammalian cells in exponential growth, yeast cells in exponential growth, bacterial cell in exponential growth. Okay, we had a question there. Okay, so don't take these numbers as the Bible, of course. But I think these are useful in order to carry some picture in your head that you can afterwards, if you have more information, you can update it with more accurate numbers. But still, you have something to start with if you want to think about the dynamics inside cells. So just to recap it, we have two different processes. For mRNA, it's more dependent on degradation. For the protein, it's more the cell cycle. And this overall time, this tau of the delay that you have, will be depending also on the organism of choice, of interest. And you also have some extra time of delay that we were calculating before, and that's for having the first few ones becoming functional. But here we're talking about how long would it take until you have, say, half of your steady state levels. And as you can tell, this would be significantly longer. Now you might also see why I was saying here, it's easy to think about as mRNAs, but if you think about it as protein, you could tell me, hey, in protein, I also need the time to make the mRNA, right? Well, that is definitely true, so you should add that in. But as you can see, because this is, if you think about the protein, because these are characteristically much longer than this, maybe you should add up this together with that, but that would not change these values by a lot. Okay, this is an example of what is known as separation of timescales, where if you have something that's very slow and something that's much faster, if you think about the slow process, there's a separation here between the two timescales, and actually you could think only about the slow process. The fast process responds so quickly that its effect on the time that the slow process takes is negligible. Okay, if that wasn't very clear, think about it again, watch the video, or just read about it. Okay, so we have some characteristic timescales. Any suggestions or comments about that? Okay, so I think we could move on. So until now, we were talking about transcription, translation, what happens in the central dogma. Now I want to take you to a different kind of question and then see how both ends, I hope, would connect together. And so question is, how long does it take to diffuse across the cell so what I'm doing here, I'm moving from the central dogma to what happens, let's say, when you have a receptor that gets bound by some ligand, and there's some process of, after phosphorylation or whatever, this has to go, say, to the nucleus or somewhere else, and this would be the thing that would trigger the central dogma in responding, and then we'll see how these two timescales talk with each other maybe later on. So in thinking about this definitely would depend strongly on what organism you're talking about and what is the relevant size. So I want to give you several options, and let's think about, say, bacteria and some mammalian cell. Again, I'm thinking here about, say, a HeLa cell. And so the options now that will make them more interesting. Let's say in bacteria, 10 microseconds, and then mammalian cells, 100 microseconds. Or in bacteria, 10 microseconds, and here 10 milliseconds. 10 milliseconds and 100 milliseconds. 10 milliseconds and 10 seconds. And reaching the end, let's say 10 seconds and 1 minute. Finally, 10 seconds and 10 minutes. Yes?
You say diffuse across the cell. Is that one molecule to go from one side to the other, or all the molecules to be evenly dispersed? Or?
So I'm thinking here that the mental picture I have in my head is, say you have a molecule, let's say some protein or something like that, that got phosphorylated at one end of the cell. And now, how long would be the characteristic time until it diffuses such that it got to a distance that's on the order of the size of the cell? Okay, you can think about it until it like become homogeneous or whatever. It's not homogeneous, but gets further apart. You were asking about the question of, let's say, if you had a lot of molecules sitting somewhere, how long would they diffuse? Okay, so the two timescales I think are actually equivalent, also in an interesting way. But if it's not intuitive to you, that's fine. Let's take this concrete example. Is the question clear? Okay, so time to cast your votes. Who's voting for A? Who's voting for B? Who's voting for C? Who's voting for D? Who's voting for E? And who's voting for F? Okay, so we have quite a diffuse crowd and a set of responses. I know, maybe I should have made more, and then I'll have one person in each possible answer. Okay, so I guess it's interesting, right? Because this is something that happens all the time, right? You have things, we're diffusing all the time. Receptors get activated. We need to convey the information on that. So let's see how are we calculating such a problem. So the governing dynamics of thinking about diffusion, it's part of what's known as random processes or random walks. And there's very elegant mathematics behind it, but I'll spare you the joys of that. Although, if you want, there's a great book by Howard Berg. Howard Berg called, I think, Random Walks in Biology. And it has pretty good explanations. It also covers a lot of ground in all sorts of examples. But I think from all of that, what you need to know is basically one equation, and that is that if we're thinking, so we were thinking about, say, a molecule sitting here, and then it starts to diffuse. Diffusion and this random walk means that you're starting to do some process of moving in all sorts of directions. And then finally, we're interested in knowing the distance r that you've reached after some time tau. What? Okay, so the question is, how am I talking about 2D or 3D? So here it's 2D. I'm thinking usually about 3D because I want to move inside the cell. But actually, in some cases, in the membrane, I might be actually also talking about 2D. There's also diffusion within the membrane. There's diffusion of transcription factors on the DNA, which is really cool. The transcription factor, after it binds, it does also some diffusion on the DNA, and that's diffusion in 1D. And luckily, it's everything I'm going to show you is almost all the same. Doesn't matter if you think about 1D, 2D, or 3D. Oh, so you'll see it's quite general. And then in thinking about this process, we have R, we have tau, and we have another player. What would be the third player? So there's something, say, about the step. So if I'm thinking about random walk that I'll be moving here, it's something about the step that I'm taking. In biology, in quantifying that, there would be something called, usually term D, that's a diffusion coefficient. We'll go back to it in a minute. But in terms of the relationships between these three properties, which is what you need to know from this book or from all of this field, is that R, the distance that you're getting, goes as the square root of the diffusion coefficient times the time. And then there's a prefactor here that is 6. That's when talking about 3D. If you're talking about 2D, it's actually 4. If you're talking about 1D, that's 2. So it's really not a big deal, as you'll see. It's basically, it goes as a square root of some small prefactor times diffusion coefficient times the time. Yes?
When we discuss diffusion in biology, shouldn't we consider the concentration of the molecule?
So you're asking, we're talking about diffusion. How come you're telling me an answer or something about an equation without talking about the concentration, right? Okay, so it's a good question. Let's see if one way to think about it is just like like in the question that was said before. Think about one molecule. Okay, if you have one molecule, then I don't need to think about concentrations anymore. I'm just interested in its random walk, right? So this, if you like, take from the perspective, the intuitive perspective, what happens to one molecule. If you want to connect it to the concentration field, this is what I've hinted at before. Surprisingly, it would also give you the same time scale and distances on the average. On the average, not the first protein that would be able to diffuse somewhere, but the average how we diffuse. Okay, and it's not trivial to see how this connects, but take my word for it or read Howardsburg words for it. Okay, so this is the basic equation that we need to know. You could also turn it on its head, which, and by the way, you see that there's an interesting characteristic. The distance that you're getting does not scale as the time. It doesn't go proportional with the time, with tau. It goes as the square root of the time. Why is that? Because if I'm doing random walk, I'm not progressing always in the same direction. Sometimes I'm moving forward, sometimes to the side, sometimes backwards, forward again. On the average, I'm not just moving linearly with time. I'm moving sublinearly. How sublinearly? As the square root of tau. This could be turned in order to see how does tau scale. So I just need to square this, and it would go as the r squared divided by 6 times d. Okay. In this course, we're interested in concrete numbers and not just in equations. So let's try and plug numbers behind that. So for that, it would be useful, first of all, to know something about, so r and tau we'll be calculating in a minute, but we need to know something about the rate of, about the diffusion coefficient. So this could be given in different ways. A good number to remember is about, say, a protein in the cytoplasm. And it would be on the order of 10 micrometers squared per second. So let's just see what I was giving you. So I'm talking about like some small protein. If it's a metabolite, by the way, if you're thinking not about a protein, but just like some small, say, a sugar molecule, this could be, say, 10-fold higher. So from 10, it would be 100. Okay? And with these two numbers, you mostly covered for anything you need. I'm talking about the cytoplasm. I'm not talking about what is measured, say, if you go to a handbook and see what happens in water, because inside the cell, things are quite different from what happens inside pure water. So usually, it's like 10-fold smaller. So actually, if this protein was in water, it would be, say, 100 microns squared per second. And in the cytoplasm, it's on the order of 10. Although, yeah, this 10 also is not coming from it's not some holy number. It's a range, but this is a good characteristic order of magnitude. And the units here you'll see are a bit funny. Usually I'm trying to stick to MKS, right, having meters. Why am I putting here micrometers? Because otherwise it's very easy to get confused, because if you're doing it, so this is 10 to the minus 6, so in meters 10 to the minus 12 would have been 10 to the minus 11, and then 10 to the minus 11 it's easy to forget if it was 10 to the minus 9 or if it was 10 to the minus 6 or 10 to the minus 13. 10 is easy to remember, and then its micron are also the relevant length scale if you think about bacteria, if you think about mammalian cells, so that would be quite useful also in the numbers following up on that. There was a question here, or not?
Did
Yeah.
anyone calculate it to diffusion across membranes, phospholipid membranes?
So first of all, anything you want was calculated in this field. There's lots and lots of biophysicists that, physicists that finished solving all the problems that they were interested in physics. They moved into biology, and then they calculate everything you can think about, definitely related to diffusion, including diffusion across membranes, within membranes, lots of good stuff about it. Much of it I don't even know, but I'm pretty sure there's good things about that. Okay, let's take all that and go back to our original question. Okay, so bacteria. This is not so good. What would be tau for bacteria? So we know the answer. It should be like r squared over 6d. What is r? So a good characteristic length scale would be 1 micron, so squared, divided by 6 times 10 micrometer squared per second, which gives me 1 over 60 of a second, which is roughly 10 milliseconds. Same thing for my, say, HeLa cell. So as a characteristic size for the HeLa cell, I'll be taking 20 microns. And then I need to square it, divided again by 6 times 10 micrometers squared per second. So I have about 400 microns squared divided by 60 microns squared per second. So that's roughly 10 seconds. So going back here, I see some people, yeah. Note, by the way, the non-intuitive nature of the fact that even though the sizes is, say, 20-fold bigger, the time scale was not 20-fold, was more like 1,000-fold. Why? Because we said it goes as the square of the distance, right? So 20-fold would turn into 400-fold. And if you do the rounding, it could even turn into 1,000-fold. This, by the way, brings up interesting questions. So a student in my group, Nivan Tonovsky, which you met last time in the calculation on the viruses, when he saw that, he said, hey, there's something interesting here. When you show me these two timescales, the timescale here and the timescale that we did just before of the time of expressing a protein. Let's say when I'm putting into my bacteria resistance gene for antibiotic. And let's assume it's a responsive gene. It's not something that's being constitutively expressed. So we know how long would it take to respond in order to produce once you're giving some, once it senses that antibiotics is there. How long? What is the relevant time scale on that? So we just calculated, right? We said there'll be a delay of about, say, a minute. And then to reach half the functional level, that depends on the degradation cell cycle, but it will be several minutes for sure. But then the antibiotics, how long would it take until it diffuses inside the cell? Milliseconds, tens of milliseconds. So there's an interesting puzzle that raises about if you're not constitutively expressing, how is the cell fine with just having it on its genome, and there would be a delay. Okay, I'm actually leaving you with that puzzle. Anyone's interested in trying to solve it, I'm actually also interested in understanding it better. So that's another option for a good final assignment. But what I want you to do now, not as a final assignment, is to now answer the following question. I'm I have the same question as here. How long does it take to diffuse across the cell? But now I want to do it, not just any cell. I want you to calculate it. I want to do it, this cell, for an axon. And let's say I don't want to be extreme, in a huge axon. Let's just think about some, let's say, 1 centimeter length. And let's give you some options also on that. A would be 1 minute. B would be 1 hour. C would be 1 day. And D would be 1 week. I have also E, 1 month.
So we assume the same diffusion coefficient?
And we're assuming the same diffusion coefficient. Yeah, that's interesting that you asked that. And that's it's actually fun, right? Because you could say maybe things are very different. Bacteria, cell line, axon. But thinking from the perspective of what we have diffusing here, we have some molecular player, some small protein. It doesn't know if it sits within a bacteria or within an axon. It's molecular microscopic details that do not know, that should be quite similar in terms of the biophysics in these different conditions. The crowding. Okay, so good question. So you're asking what happens about the crowding. So if you look at the cell dry content within cells, it's all pretty dense. That's why the diffusion coefficient should be taken in cytoplasm, not in pure water. But it's roughly the same concentration in bacteria as in cells in our body. Let's say 2/3 water is a good. The contact, the interactions. So the interactions are also quite, grosso modo, I'll say they're similar. Of course, there are specific details that could affect that. Okay, so who's voting for 1 minute for the axon, the time to diffuse across a 1 centimeter axon? 1 hour? 1 day? Half the class? 1 week? 1 month? Okay, so for that, I don't want you to take 1 day, but do take 5 minutes and try each one by yourself to calculate it. Okay? Starting now. Maybe we'll make the votes once again. Who's voting for A after doing the calculation? B? Silence. C? D? E? Okay, let's just try and do it ourselves. So we have this governing equation that we said the time goes as the R squared divided by 6 times the diffusion coefficient. Let's plug in the numbers once again, just like we did here. So tau for the axon. So what is my R? 1 centimeter. So 1 centimeter, I want to do it in micrometers. So I have 1 centimeter times 10,000 centimeters per, 10,000 micrometers per 1 centimeter. Squared divided by 6 times 10 micrometers squared per second. This was just a fancy way of doing it, just like as we did before, that we're always allowed to multiply by 1, and 1 centimeter is 10 micrometer cubed, right? 10 micrometer. So I have 10 to the 4 squared, so I have 10 to the 4 squared, that would be micrometer squared, divided by 60 micrometers squared per second. So it's 10 to the 8 micrometer squared divided by 60 micrometer squared per second. So it's 10 to the 8, let's say 10 to the 2, so I have about 10 to the 6 seconds. So, okay, let's talk about it in just 1 second. Let's just finish with that for a second. So you might remember we said about, say, seconds in a day, about 10 to the 5. So we're talking here about 2 weeks. Okay, so it's somewhere between, say, D and E, if you want to move 1 centimeter. Okay, if you want to move 1 meter, you can try and play that out as well. What would happen with the scaling? So 10 meter versus 10 centimeters, that's how much more? 100-fold more, right? But we know that the time goes as the square. It's not 100-fold more, but 100 squared, that's 10,000. So 10,000 weeks, like 20,000 weeks. That's a lot of time, right? 20,000 weeks, that's like 300 years or something like that. So there's hopefully a different process that would enable things to move. And actually, they have to, right? Because, say, the mRNA, let's say, it gets produced in the nucleus. If it needs to reach the end of the axon, you need to find a different trick, right? And there are other tricks. For example, you could move using a molecular motor, or maybe you could have it produced in different ways. There's actually a lab here of Mike Feinzilber that actually studies all sorts of new tricks that science was not expecting of how that could be achieved. About the question that was raised before, about whether we should use here an equation for 1D rather than 3D, I'll just say that, there's different considerations. In the end, it doesn't matter, right? Because if I have a factor of 2 or factor of 6, it's a factor of 3. So maybe it would take you just a little bit less, although we could consider which one is the right answer. Comments on that? Okay, so now I think it's a good time to try and put these two things together. And the way I suggest to do that is the following. So a few months back, when I was just starting to think about this course, I was driving to Jerusalem with Hila Gingold, to a conference in Jerusalem with Hila Gingold, who's a very smart student from Tzachi Pilpel's lab. And she was telling me about the following idea that she had when she started her PhD. She said, consider an mRNA, and let's think about what happens with, you have a codon here. This is codon I. And let's say it codes for amino acid I. Let's think about what happens when you have an adjacent codon that's pretty close. Let's say it's sitting here. Let's say it's quite close. Let's say it's just 2 codons further apart. So that's codon I plus 2. It would code for amino acid I plus 2. Now, very often you would have, not very often, but it definitely could happen, that amino acid I and amino acid I plus 2 are the same. That you need the same amino acid soon after. And the question was, would it be beneficial for the cell to use the same codons when you have something in local proximity, to use the same codon rather than a synonymous codon that is being recognized by a different tRNA? Okay, so the idea was, does 2 adjacent codons translate faster when coded by same tRNA due to local concentration enhancement. Okay, so what is the core of the idea? It's thinking, how can we optimize, thinking how could cells optimize their functioning? Because definitely there's been lots of time for evolution to play around and try to optimize things. So let's try and see what are the implications of that and then check if we could write a paper on that, right? So the question is whether you already have a tRNA that was bound to and got to this codon and put some amino acid. If you need the amino acid quite close once again, the tRNA would probably not get very far. In that respect, it could come up again and be used again. So it might be better to use the same type of codon rather than a synonymous codon. Okay? The idea is clear? And luckily, today it's not so difficult to try and study these things because we have, lots and lots of genomes. For all those genomes, we know exactly how the different sequences then translate into the amino acids being used, into the codons being used, what tRNAs you have. So you can do that bioinformatically and then test on that. So even though that was Hila's idea, she started working about it, but she got distracted with some other projects in the lab, and she didn't finish working on that. And that was somewhat disappointing when she found not long after that there was actually a paper in Cell that was talking about exactly this sort of dynamics. So let's try and analyze that and see what are the timescales and how that could work. So I'm interested to know what would be the implication in terms of this local enhancement concept, right? So let's say you had the tRNA, it came there, it put its amino acid, and now it needs to reload and put another amino acid once again. So there's some time scale that it would take, some tau, that it would take to go one step further in the translation process. So what is that time scale, roughly? Until it would put the adjacent amino acid. Right. So it's related to exactly the time of polymerase. So we said that the velocity of the ribosome is roughly what? About 10 per second, 10 amino acids per second. Right? So that means that the time scale for the adjacent, for putting the adjacent, so let's say maybe it just left here and then maybe until you need to get to not to the next one, but to the one after that, that would be on the order of, say, 0.1 second as a result of that. Maybe it's 0.2, maybe it's 0.1, depending on what exactly do you want to take in the process of how long of the step is it until it unbinds and then binds again, or something like that. Okay, but this is the characteristic time scale. Okay. So now all we need to know is, I'm interested to see how far would the tRNA diffuse in the time scale of this 0.1 second after it was released and until it will get bound again. So what is the distance that it goes? So this is the original equation, so I'm flipping this one again on its head. it goes as square root of 6 times d times tau. Tau, I know it's about 0.1 second. D, we said about 10 micrometers squared per second. So we said that's for some protein. Here we're talking about tRNA. So what do we know about tRNAs? tRNAs, if I remember correctly, about like 70 nucleotides or something like that. 70 nucleotides, so this nucleotide is like 3 times the mass of an amino acid. So that's something like equivalent in terms of mass to like 200 or 300 amino acids. So it's roughly the same size, and therefore I could take roughly the same diffusion coefficient. So I have square root of 6 times 10 micrometers squared per second times 0.1 second. So that's 60 times 0.1. So that's the square root of 6 micrometers squared. So that's about 2 or 3 micrometers. So, and I forgot to say that, so the analysis, so Hila is studying mostly yeast cells, and the analysis was done for yeast as well as bacteria, although it could be thought about mammalian cells as well. So let's think about it. In the 0.1 second from the unloading until it would need to reload again, it would diffuse about 2 microns. So if you're a bacteria in 2 microns, you've covered the whole, like you've reached the other end. Also in yeast, you've covered, like, say, a very significant fraction of the overall volume. Meaning that in terms of thinking about what happened to the local concentration, there's hardly a blip on the local concentration of that specific tRNA. It's just like any other tRNA, given what is the concentration of that tRNA inside the cell. There is hardly no enhancement if you look at it quantitatively, not if you look at it qualitatively, right? I could hand wave the argument quite easily about why it's great and very optimal, but actually when you just put the numbers and see what happens there, it's actually a negligible effect. Okay?
Also, if you look at it in the DNA level of the sequence, you might not want that specific repetitive sequences, one very close to another, because that could give you totally different mechanisms.
Okay, so so there could be all sorts of issues relating what do you prefer and whether that could be detrimental if you have very similar sequences next to each other. That's a different thing. But let's go back to this. So what about the paper, right? So I was quite confused about it as well because, you know, it's like these cell papers. So cell papers, I almost never reach the end. They're like really long and there's lots and lots of figures. But actually when you reach the discussion, they're actually considering, so they're not doing this type of calculation, but they were suggesting that maybe there's a mechanism behind it where the tRNA actually does not gets lost and starts to diffuse around, but actually is tethered into this big complex of the ribosome together with the mRNA, and you have these tRNAs still being tethered and have some protein-protein interactions, and therefore it's not like it starts to diffuse. It stays next to the ribosome, then reloads with a synthetase that puts a new amino acid, and then could come and reload again. Okay, so under that mechanism, that could explain it. But it's not the naive way in which the idea could be thought about and how actually I read the paper originally. Yes?
How long does it take to reload an amino acid to a tRNA? Like, is it the same time scale?
Okay, so
Relevant.
okay, so the question is, how long does it take for a tRNA to reload an amino acid? That's a good question for a final assignment.
Would you say that to justify such a tethering mechanism, you have to see relative abundance of repeats of amino acids that are more significant than would be expected from random?
Yes, definitely. Okay, so what is being said is when doing this analysis, there's all sorts of, let's say, normal bioinformatic caveats. You need to take into account what is the propensity for using different amino acids, what is the propensity for using different codons, for doing this, like correcting for AT and GC biases and all sorts of things like that. So this was analyzed to death, right? There's like several figures when they're normalizing for that. They're taking only, they're swapping the DNA content in that specific organism, only in coding sequences, only in promoter sequences, only in exons. All of those corrections were done very heavily. But then there's a different way of thinking about it, not from these sort of biases, but just from plugging in the specific numbers and seeing what would they imply on the process. And this is sort of like the thing I want you to come up with. Okay, I think this is enough for rates today. Looking forward to getting your subjects for assignments and seeing you next week for information and errors. Thank you.